In this lecture, we'll focus on irrational design of peptides, and predominantly the technique used is solid-phase peptide synthesis. So in solid-phase peptide synthesis, or SPPS, reactants are bound to polymeric surface and modified while attached. Final product is released at the end of synthesis. The advantages are that you have specific reactants that can be bound to specific beads. Beads can be mixed and reacted in the same vessel. Products formed are distincted for each bead, physically distinct. Excess reagents can be used to drive reactions to completion. Excess reagents and byproducts are easily removed. Reaction intermediates are attached to the bead and do not need to be isolated and purified. Individual beads can be separated to isolate individual products. Polymeric support can be regenerated and reused after cleavage of the product, and then automation is possible. In order to make libraries of peptides, you can use combinatorial peptide synthesis. It combines aspect of combinatorial chemistry, which allows one to create a number of different compounds simultaneously. And instead of just making compounds, we're focusing on peptides. So combinatorial peptide synthesis allows one to create a number of different peptides simultaneously. And in order to do this, you take advantage of SPPS. Theoretically, the number of possible different individual compounds, N, prepared by an ideal combinatorial synthesis is determined by two factors. One, the number of blocks available for each step, which we can call B, and the number of synthetic steps in a reaction scheme, which we can call X. And so the equation that we're looking at in terms of determining the theoretical possible compounds, so N would equal B to the X factor. So if we look at this equation and then look at library calculation shown here, for a basis set of 20, which is the 20 amino acids, if we're looking at polypeptides, right, that value B is the number of blocks, so 20. So that would be 20. And then the number of synthetic steps would be for a peptide that are 3 amino acid will require 3 synthetic steps, would be to the X, or the third power, and so that would give you the value of N, in this case 8,000. As you increase the number of peptides or amino acids in your library, that number increases dramatically. You can have even larger basis sets, so non-natural amino acids, which increase the set. And then the same equation is as follows, where you have, if it's a 3 peptide with non-natural amino acids, you calculate all the values shown here. So in terms of peptide libraries on solid support, I'm going to be talking about 5 different methods. One is parallel synthesis, second is split and mix synthesis, reagent mixture synthesis, multipin Pepscan, and teabag method. In automated solid-phase peptide synthesis, or just solid-phase peptide synthesis, the common solid support are beads. The beads must be able to swell in the solvent used and remain stable, and most reactions occur in the bead interior. So here's a resin bead, which is essentially a polymer we had talked about in the previous lecture. You swell that polymer, exposing the sites for linkage for the reagents to covalently attach, which allows for synthesis. In terms of parallel synthesis, you can use solid-phase peptide synthesis. Parallel synthesis peptide libraries are synthesized in individual chamber reactions, shown here. The benefit is that each product is pure, separated, and well determined. The identity of each structure is known. The con for this is that only a small set can be generated as a peptide library, because you're limited by your reaction chambers and how you can add all the reagents per reaction chamber. So for this to be used to make large libraries, you need an automated synthesis system for large pools for peptide libraries. So here's an example of automated parallel synthesis. This essentially is a peptide synthesizer, which allows for the synthesis of large pools of peptide library. Usually has a multi-channel head, and then wells, tiny wells that can pick up reagents and then deliver reagents to each well. Automated synthesizers are available with 42, 96, or 144 reaction vessels or wells, shown here. They use beads or pins for solid support. Here's an example of pins for solid support. Reactions and workups are carried out automatically. Same synthetic route used for each vessel, but different reagents. And then different products is obtained per vessel. So each vessel will have a different chemical product. So it can be possible, since you're attaching you have pins, your beads attached to this top support. You can then have these wells contain the reagents and then dip your bead so that the reagents for particular amino acids can be attached, and then continue to dip to other amino acids so that you can get a different polypeptide. And then you can take these beads, subject it into cleavage into new reaction vessels, and then that polypeptide will be released into those reaction vessels. So if we look at automated parallel synthesis, you can generate 27 tripeptides from 3 amino acids. So let's look at your beads. So you can take a bead, and then each bead can have a different amino acid. 2 of the same amino acids are being shown here. After you conjugate that one amino acid, you go through steps to then add another amino acid. You go through another series of steps to add a third amino acid, and then you can cleave it off. So as I had said, for calculating the diversity of this library, you have a basis set of 3, and you're making a tripeptide, so that goes 3 to the third, and so then your solution is 27. In automated parallel synthesis, you're using standard SPPS. So in order to get those 27 tripeptides, you're going to need 27 vials, because each vial will signify the tripeptide, the potential tripeptide. Split and mix synthesis is a different strategy that is used to generate large libraries. To use a standard synthetic route to produce a variety of different analogs where each reaction vessel or tube contains a mixture of products. So that's what split and mix synthesis generates. And the advantages is that it's useful for finding a lead compound. You're capable of synthesizing a large number of compounds very quickly, and we'll show you in the next slide how this is possible. But the con for this is that the identities of the structures of each vessels are not known with certainty, as in the case of parallel synthesis. Split and mix synthesis example is shown here. You want to synthesize all possible dipeptides using 5 amino acids. If you're using the standard parallel method, you would need 25 separate syntheses. In the case of split and mix, you would involve 5 separate syntheses using a mix and split strategy. So this shows what a split and mix synthesis is. So here are your beads, and then you attach your amino acids to the beads, and you get a solution of these 5 different amino acids being attached to the bead in a single solution. So now you've already combined them. Then what you do is split them into 5 equal reaction vessels, and then you add your amino acid to that, the 5 different amino acids, and so that you get a mixture of your all possible combinations, leading to the 25 possibilities. And so in this case, instead of having 25 separate syntheses, you have just 2 syntheses happening, because you're doing one reaction, right, shown here, but you're combining them. Then you're splitting them, and then you're doing a second reaction. Split and mix synthesis can provide equimolar mixture of random and large peptide libraries. And there are essentially 3 processes involved in split and mix synthesis. First is splitting, then it's coupling, and then it's mixing. So here you can see you're splitting, coupling, and then mixing at each step shown here. And each bead that exists in the mixture contains one peptide. And so there's one bead, one compound. For simple, short peptides, it's easy, at least easier to determine what your peptide library is. However, as you get to longer peptides, decoding and knowing what you have in your peptide or in your library is important. So how does one do that? You can use general methods, LCMS. You can add a DNA tag to the bead simultaneously as you couple with another amino acid, and then use PCR to amplify and identify. You can use chemically inert tags that can be identified by GC, or you can use optical barcodes. So you can use LCMS, Edmond degradation, but this is time-consuming and expensive. Another approach is to use ladder peptide synthesis, where small portions of peptides are N-terminally capped after each coupling. Therefore, peptide ladders are released and analyzed by MS. So you can, so these would be the capped peptides after each coupling. And so when you cleave this off and you view it under MS, you can then get a ladder and identify what that sequence is. You can co-synthesize a tag, in this case a DNA tag, so that for each amino acid you're adding a DNA base. Each combination will have a different nucleotide base that can be readily read by PCR amplification. And so in this case, as you're adding your building block, you're also adding your DNA tag, so that on your bead you have both your peptide or your amino acid and then your DNA. You pool it, you do the same thing, now adding your amino acids with their respective tags, so that you're adding your amino acids and also adding your nucleic acids. And you can pool again and do the same thing. And so for each bead, you get your peptide and then your DNA tag. The advantage of this is that you don't need to have large amounts of DNA, since you can PCR amplify, and that's good. But the cons for this is that the tags are chemically labile, so the DNA tags aren't as stable, and they're incompatible. And another con might be that the oligonucleotides may interfere with the assays that you're looking at, because if you're looking at more biological assays that involve proteins and other macromolecules, it might interfere. Chemically inert tags can be employed to decode what's on your bead, and if you make them photosensitive, you can then use UV light to trigger its release. So here's an example on your bead of a chemically inert tag, and it has a photocleavable link that upon UV light will release the tag. that allows you to identify, at least the tag, based on tag, what the polypeptide sequence is. And so as you're generating your polypeptide, you're adding specific tag molecules to your bead based on your tag. And then when you release it with UV light, you're going to just get your tag molecules that you can put under GC, and then you can look at their retention times, and based on their retention times, identify whether the tags represent this amino acid or that amino acid or that amino acid. So barcodes have been developed, and barcodes use quantidot cadmium selenide encased zinc sulfide shell, which allows it to be fluorescent. And so these fluorescent crystals are used as a tag for each amino acid. And depending on the size of the quantum dot, you can get different fluorescence. The ones in these digital codes are represented by a peak in the visible spectrum, and the zeros by an absence of the peak. And so shown here for a bead that represents a particular amino acid, you're going to see 1 here, but 0 and 0 because the others aren't shown. For another amino acid that has these 3 quantum dots, you'll get 3 peaks in the emission spectra, which will lead to a spectral code of 111. These quantum dots are actually encapsulated in the bead interior during the bead synthesis of the amino acids on the bead, so that they're part of the bead. So you don't have to use UV light to remove the beads. It's actually part of the UV light to remove the tags, I mean. It's actually part of the bead, and it's in the bead. So by just scanning it on a fluorescent microscope, you should be able to identify at the particular bead what that sequence of polypeptide is. In practice, because the emission peak of a given dot is so narrow, it is possible to squeeze 6 different resolvable peaks from 6 different sized dots into the visible spectrum. And the number of digital codes these dots could generate is 2 to the 6 minus 1, or 63. And so that is determined, that this number shown here can be determined by this equation. So I'm going to draw it over here. C being the unique number of codes, N being the intensity level, M to the M being the emission of color, the emission color, minus 1 is what is being used to calculate this below. So while this number is low, you can actually change the intensity of the light emitted by the dot, and you can vary that into at least 6 steps. So if you do that, then this number becomes higher. So instead of having 2 intensities, you have 6, so it would be 6 to the 6th power minus 1, which would lead to 46,655 codes. In reagent mixture synthesis, you take all your amino acids and you add them in one reaction chamber vessel and allow them to react based on their isokinetic ratio so that a particular sequence is generated. So while the advantage of this is that you can just add all of your amino acids and in a single reaction vessel with the aim of generating a peptide sequence, the disadvantage is that you get more than one peptide per bead because you can get inefficient coupling happening, and so it's not going to be one bead per compound. Here, isokinetic ratios are calculated and the corresponding amounts of each amino acid reagent employed in the coupling reaction. The Multipin technology involves synthesizing peptides onto specifically prepared polyethylene pins, shown here, mounted on blocks in a format which is compatible with the standard 8 to 12 microtiter plate used for biological assays. Both pin-bound and solution phase peptides, that means those that have been cleaved off, so this would be cleaved and this would be cleaved and captured onto plates, can be produced by this method. So these multipins that are arrayed on these microtiter plates can remain covalently bound to the pin or cleaved off, as shown here when they're cleaved off. Peptide loading on pins are about 2 micromole per pin, and it allows for the simultaneous synthesis and screening of large numbers of peptides. Using multi-pin synthesis, one can do PepScan. So you can identify peptide leads of biological interest from scanning through protein sequences. So if we look at this target antigen, you can generate overlapping peptide sequences that span this whole sequence of the antigen and synthesize them, and then identify regions of the antigen sequence that bind to the antibody, even though they're not continuous. So, for example, if this target antigen is folded in some way, and this region and this region are close together and recognize the antibody, you can then get hits from here and here that show a non-continuous binding. The advantage is that you can wash off the antibody and the pins can be reused. The disadvantage is that it may not be consistently the same concentration. It's harder to scan the pins in solutions for antibody binding. Biotinylated peptides allow for the immobilization on surfaces by using streptavidin, which can be coated onto solid surfaces. And so using this, you have your streptavidin and then your biotinylated peptides. You can bind your streptavidin to the biotinylated peptides, which then can attach onto the surface. And then use antibodies to recognize the peptide and identify, using ELISA, the binding of the antibody to the peptide. And using this, you can map continuous epitopes using overlapping peptides. Teabag method, use a resin sealed inside of a porous propylene bag. So it looks like a tea bag. Tea bag contains beads and it's labeled. Separate reactions are carried out on each tea bag. You combine tea bags for common reactions or workup procedures. A single product is synthesized within each tea bag, and different products are formed in different tea bags. The advantage of this is you can combine tea bags for workup. They're cheap, and it's possible for any lab. The disadvantage is that it's a manual procedure, and it's not suitable for producing large quantities of different products. But at least for peptide libraries without a peptide synthesizer, this can be very effective. Strategies for peptide libraries, we discussed PepScan in terms of multipins PepScan. Positional scanning is something that we'll discuss. Peptide analysis from solid support and non-traditional supports. As we've already discussed PepScan, I don't need to discuss that further, so we'll look at positional scanning to start. For positional scanning, both reagent mix and split and mix synthesis are useful. Positional scanning libraries are composed of systematically arranged mixtures where you can have single position defined positional scanning libraries. Here, each compound present in a given mixture has a common individual building block at a given position, while the remaining positions are composed of mixtures of all building blocks used to prepare the library. A common single building block defines each relevant mixture. So if we look at hexapeptide positional scanning library. The zeros, or the O's, represent the amino acid that doesn't vary, and then the X's represent just the varied amino acid. So if we look at this sub-library, in this sub-library you can have a position that doesn't vary, in the position that doesn't vary, one amino acid, and then a random set, another amino acid, and randomized. So here you're having this systematically an amino acid that you know, and then the rest is being varied. Various solid supports can be used. We're going to talk about non-traditional solid support, or cellulose or cotton. You can use a disc of cotton as a solid support for peptides, and you can stack them on top of one another after labeling them with ink and then subjecting them to a continuous flow column, which I'll show you in the next slide. But essentially, on cotton, you can add a linker and add amino acids onto, after adding FMOC, your FMOC linker, and you can add amino acids where you have on discs particular amino acids being added, or particular discs being removed or added, to generate different libraries where each disc is a different peptide library member. So here is your column, and what you can do is you can put your discs in the column and subject them to washing and adding particular linkers and amino acids. So the loading is 1 to 3 micromole per centimeter cubed. Use porous polar. The porous polar nature of the support helps suppress unwanted non-specific protein binding effects. And you can see in a system, you have your cotton fabric, you wash and dry, you cut out discs, you activate it, attach your handle, acetylate label with silver ink, and then attach your linker. Once you do that, you say if the amino acid to be coupled identical for all sequences. If it's yes, you apply the coupling cycle. If it's not, you sort the disc. More than 3 identical amino acid is coming in the coming sequence position. If it's yes, you apply coupling cycle. If it's no, you remove. In the FMOC, you sort the discs, you attach amino acids in beakers, combine the discs, and coupling cycle. And so you go through this process so that you generate different discs with different peptide libraries. Another non-traditional support is peptide microarray. There are 2 types of peptide arrays according to their preparation method. One is immobilization of pre-synthesized peptide derivatives. So essentially you generate your peptide libraries either probably using standard solid phase peptide synthesis in parallel, and then you add those pre-synthesized peptides onto your substrate, or your, in this case shown here. Or you can do in situ synthesis of peptides directly from the surface. For immobilization of pre-synthesized peptides, you need chemoselective immobilization methods, and this is important for controlling the orientation and the density of the immobilized peptides. So the solid supports that can be used are glass, polymer, chips, and the critical factor is the accessibility of the peptide to the target. So you must need to add linkers and spacers between peptide and chips. So oftentimes, as you've seen in the previous slide, things like PEG are used as linkers. So here are some examples. You're using aldehyde to do amine and amine reduction to add. You can use reduction with amino oxyacetal groups as in this case. You can do cysteine residue, an N-terminal cysteine residue, and react it to thioester linkers to generate your peptide being attached. And then here's cyclopentadiene reacting with benzoquinone in a cycloaddition to attach your peptide. While we've been discussing linear peptides, you can also generate libraries of cyclic peptides. As we discussed before, cyclic peptides are important. So in cyclic peptides, you can actually have different types of immobilization strategies. One is to generate your cyclic peptides and then attach it, and use the previous strategies to attach those peptides onto the surface. Or you can generate a cyclic scaffold, which have, in this case, 3 chemically orthogonal groups, shown here. And so you can selectively remove each of these groups, and then by, in this case, UV irradiation, and then attach your amino acid or your peptide to the selectively unlabeled or deprotected site. So here, if we look at the synthetic cycle, your NPPOC group is selectively removed by UV irradiation to expose the amine at the desired position, shown here. Activated carboxylic acid is coupled to the newly exposed amine, shown here. One chemically orthogonal protective group is removed to expose free amine, shown here. And then the free amine is protected with NPPOC, shown here again. And then you can deprotect, reacted with another carboxylic acid that's been activated, and proceed on. In terms of in situ synthesis, parallel synthesis of peptides, there are 2 major types. One is spot synthesis, and the other is photolithographic synthesis. In spot synthesis, here you're using a cellulose membrane. You can also use polypropylene and glass as other substrates, but your peptides are synthesized in a stepwise manner on that substrate. So in this case, on your membrane, you spot on a spacer and a linker attachment at a particular spot. And then on top of that spot, you start to synthesize or add amino acids. So in this whole process, you have small volumes containing activated amino acids that you spot on the substrate. Each spot is a microreactor composed of delivered droplets, and you employ solvent that is non-volatile to maintain the wet state. You deprotect and cleave or bioassay. So after you've added on, you can either deprotect and cleave, or you can deprotect and then right on that membrane do an assay. In photolithographic synthesis, you're using deprotection strategies that use UV light to deprotect a particular site, and you use masks to keep the protection on particular sites. So here's a mask in which you use UV light that can deprotect this particular site. So you do photo deprotection, then you add a reagent that can chemically couple, and only the sites that have been deprotected can be chemically coupled. You can then use another mask to deprotect the other site, chemically couple again, so that at a different region you have different chemically coupled amino acid, and you can repeat so that you have libraries that at different positions have different sequences. In photolithographic synthesis, patterns are generated using masks. So you can generate a pattern based on orthogonal stripes, so that you have all of A's, all of B's, all of C's being laid down, and then all of E's, F's, and G's. And so in this case, you would get 4, right? Because there are 4 sets of building blocks to the 2 power, so 16 compounds, 8 chemical steps. You can also use binary synthesis, in which you use compound A being generated, laid down on half this, compound B on this half, compound C is stripes over here, and compound D is stripes over here. When you're doing this, you're using 2 to the 4, and using this, you generate the same number of compounds using half the number of steps. You can then conduct maskless photolithographic synthesis with advances of optics, and so that you can actually control where the UV is being placed on the surface of your chip, and using microfluidics to deliver reagents and wash so that you can generate polypeptides very easily without using a mask on the surface. So in summary, we've discussed encoded mixture synthesis using either nucleotides, peptides, or other chemical tags, and even fluorescent tags using quantum dots. And the active compound is identified by its tag. We've talked about arrayed spatially addressable synthesis, where the active compounds are identified by molecular weight, components, or location. We've talked about deconvolution, trying to identify what's on those beads, and screening. So using these techniques, you can have different applications. You can do peptide ligand screening, which we talked about, antibody binding, epitope mapping. You could do enzyme substrate and inhibitor screening, as well as peptide catalyst screening. So in terms of identifying binders to antibodies, you can label the peptide with a fluorescent tag, and you can directly monitor those that are bound. So here are naturally loaded peptides with specific antibodies. You can then use high affinity fluorescently labeled peptides, and those that are fluorescently labeled you can count. You can also add test peptides to that and then see how it effectively competes off the ones that are labeled. This would be a direct strategy of detection. An indirect strategy would be labeling a bead with a tag that can detect what's happening in terms of a catalyst reaction. So here is your peptide, and bound to that bead is this moiety shown here, which upon substrate, right, addition, if your peptide can catalyze that substrate, it will produce acid, and that acid will then add to this moiety, allowing it to be fluorescent, and then you'll be able to identify that fluorescent bead. So while these strategies for generating peptides on solid support allows one to generate libraries and then scan those libraries and sequences for function, you always need to rescreen to test for false positives. So if we look at solid support peptides, you have multivalency because that is due to the fact that more than one peptide is attached. So if we look at, for example, this region, you have more than one peptide being attached, and so you have multiple peptides that can help bind to your protein or macromolecule of interest, and that might lead to something that's a false positive because you have so many of these being attached. So in order to confirm what's going on, instead of having one do the assay again on solid support, you want to confirm in solution. So you want to have these peptides in solution, and then see if the binding is actually indeed true. And so while the screen is important, confirming the screen in solution is equally important to make sure that it's valid.